So thanks to all of you for attending and those online. I will try and use the laser pointer on screen versus a physical one. So I might be looking here for the benefit of those online. I thank Olivia. She was very gracious in trying to get me to come here and forcing me to come here. But I've had a wonderful time. Really like Georgia. So live in Michigan now, and we won the national championship, so I thought I should just put it out there. Los Alamos, it's a great place for general research. You may have been introduced, maybe a bit more recently via a movie that recently came out. I got to see it over the holidays. It was great. And if you still don't know about a little bit or anything about Los Alamos, I can introduce you. It's one of 17 or so national labs that are throughout the US. It's the largest national lab. We also have the largest postdoctoral associate population. We are very large in surface area and number. So we have about 14,000 employees. The town of Los Alamos is about 18,000. So you can imagine some of the conversations that go on at various drugstores or while you're shopping for vegetables. We have a very diverse workforce, or at least for New Mexico. Of course, our biology or bioscience division is even more diverse, but this is totals for the entire lab. Our budget is not 4 billion. That would be wonderful. We could do a lot of that. It's very lovely. Like I mentioned, we do have a lot of postdocs, and we're always interested in hiring new ones. We had Olivia, who obtained a DOE fellowship grant to, which required her to do an internship at a national lab. And we were fortunate enough to have her in a different group. And then she wanted to come back with something maybe a bit more aligned with her project, since we work on bacteria and fungi. So the national lab footprint, Los Alamos is quite large. It's about the same size as Washington, DC. You have a lot of different groups, the majority of which is focused on weapons research or global security, national security type missions. We have a smaller department on science and tech, and we reside in this life sciences group. So we're actually a very minute component of this very large lab. But we've been doing biology since almost the beginning of the lab, or at least after the war. And that was to really study the radiation impact on the human genome. So that was the guise under which Los Alamos began their biology research and began the Human Genome Program. So flow cytometry was also developed at Los Alamos and was used to do sorting of the chromosomes for the Genome Project. We were also one of the initiating national labs for the Joint Genome Institute, which is the nation's premier environmental science genomics genome center. We've probably completed more microbial genomes than any other group on the planet. And then we have a number of other things that are related to genomics, including being the site at which GenBank began. Before it was moved to Bethesda. So we started a number of things on the bioinformatics front and host a number of relevant databases to this day. The HIV database is probably the most popular. And we've developed a number of tools just to make it easier for biologists to process and analyze this new flood of genomic data. Because of our position as a national lab and our focus on national security, we were also engaged or called upon to act during the pandemic. And so we were part of a number of different standards groups. And I'll try and run through a few of those, but many people don't know there's biology at Los Alamos, but we kind of shoehorn in any type of biology under these 6 different areas, and anything and everything is essentially, you can relate back to national security. And so there's a way to be able to do almost any type of research that we actually want to do, although we are called upon often to perform under specific things. Another nice thing about national labs are the tools, the fun equipment that we have at the lab. I'm only putting up some of those relevant to genomics and bioinformatics, but we have a lot of microscopy and electron microscopes there. We have a lot of other instrumentation and lab automation. microfluidics and a lot of processing or additive manufacturing going on at the lab as well. And I might show you some of the devices that we work with there. So during the, I was telling at least one of the individuals I was with today that we actually started responding to the pandemic in December of 2019. As soon as the first genome was released, we were asked to start getting involved more under a national security banner, but then eventually this became a public health emergency in the US, and we tried to respond before, even before it got to US soil. There have been a lot of work in SARS-CoV-2 genomics and has obviously ushered in this new era of genomics towards surveillance or towards pandemic assessment and towards epidemiology. There's more than 13.5 million genomes now. I apologize, but all my slides, at least on the infectious disease side, are really old. But I didn't have time to make any new ones. So, you know, we, along with many others, have contributed to the sequencing of various isolates that we've gotten from clinical samples and depositing them in some of the databases. And we've also done downstream analytics for all the genomes that were generated. The US response, although touted as really good, and the national or global response was amazing, but there were many gaps that were identified in this process. And, you know, even when the US started to respond, which was maybe a year late, we had a number of genomes available and we were starting to do accurate surveillance, but there was a call for more and more genomics because that might help us both mitigate some of the spread as well as respond a bit more rapidly to some of the new strains that were surfacing. And this was how things were done when there were only about 1 million genomes. It's a good way not to approach things in a standardized fashion. So we were starting to use a whole bunch of different methods and different tools to tackle this organism, but that, you know, everyone used whatever they had at their disposal. I'll talk a lot more on the bioinformatics component, but there was a lot of differences in the way samples were processed, in the way metadata was collected. So everything led to very challenging response because you'd have to try and shoehorn in different types of metadata into different fields from different groups. And no one was very consistent in how they were calling it. So that was a little bit challenging. The CDC had a big call. They had 7 centers generating data from different areas around the country. They had partners with different types of pharmacies that were collecting the samples from the nasal swabs that we all have done. And then they were sending this off to some of the labs. They down selected from there, did a bunch of genomics. They had many different platforms that were used to do the sequencing, many different assembly or consensus generation methods, and many different types of outputs that the CDC had to deal with. And we had done an early response to this before they started the national sequencing program. We have a number of engagements abroad as well as in country to help with bioinformatic data analysis. And as soon as the pandemic started and was spreading throughout Europe before it reached the US, we were asked by a number of groups if we could help them come up with a solution to process some of the samples rapidly in an easy way, because most of the public health responders, they have very little knowledge in genomics or sequence analysis, and they don't have time to deal with that since they have to run around with policy and with other people to get things done. So we generated this, what we wanted to and hoped to be a one-stop shop for processing SARS-CoV-2 data. And so it's a simple to use user interface with a sophisticated backend that does a lot of the analytics that you would expect to be done. It's done in a consistent fashion, and then we just added and tacked on additional reporting or additional visualizations as time went on. So the platform's been around for quite some time. We had presented it early on to the SPHERES consortium, the SARS-CoV-2 working group that the CDC was running. And they had engaged us relatively early to see if we could help out with their national surveillance system. So this is just a small picture of the workflow itself, but we just take raw data as input. It doesn't matter if it's MinION or Oxford Nanopore or PacBio or Illumina data. We do some routine quality control and processing. We do alignment against the genome, which turns out to be one of the better ways to do the assessment, although we can also allow independent de novo assembly. It just doesn't happen to work well on amplicon data. It does great on shotgun data. And then does a number of SNP calling steps and then provides the lineage by referencing the Pango group's effort in defining lineages. And we partnered with other groups like the Usher group to place it phylogenetically. And then we also allow a single push button submission system as long as you have all the metadata input into the system as well. So we had to test this early. And so when there were only a few thousand or tens of thousands of genomes, we did some analyses to see whether or not we could recapitulate the genomes that were being pushed out to the open source domain or to GISAID or to GenBank using the raw data. Unfortunately, there's no way to link raw data, even in NCBI, to the completed genome. They were not cross-linked, and so you had to do a whole bunch of metadata searches and trying to link one data set to another. And there was zero ability, even though there's a field in GISAID that is sometimes filled out on how to link it back to the raw data. It was rarely, if ever, used. And so we had to try and figure out what data sets corresponded to one another. When we did, then we could analyze those raw data, compare it to the rest of the genome that was deposited in, and then calculate issues. And we were really hopeful that there'd be very few issues that we would find, that everything was identical. But then we had to come up with a schema to be able to articulate the differences that were between what we find and what other groups find, or what's reported there. So in the perfect world, everything would have matched up on this red line with no differences between the genome that we had come up with and the genomes that were deposited. But here you'll see there's a number of off-target or different calls. And we started having to manually investigate all of these differences to see why that may be. Unfortunately, most of the time, the methods used in getting to the consensus genome are never reported. Most of the time, but not never. They are reported in some of the cases, although the details leave a little to be desired. There's no parameters. Sometimes the tools are mentioned, other times they're not. And most of the time, just nothing is mentioned. Here's the consensus and you should trust it. So here's an example, and we developed this visualization format just so that we could easily see. Ours is called the Edge COVID-19 workflow. It's based on another software system that we had developed previously called Edge Bioinformatics, which has many tools that you'd normally use for almost any type of analysis, phylogenetic, primer design, et cetera. We just grabbed that overarching system and used it for this COVID-19 work. But here we're reporting the SNPs that we find. Here's the underlying read data. If you're familiar with the IGV view, you can see that these are clearly mutations with respect to the Wuhan reference that we were using for reference. Here is we call this vendor, but that's because we were doing this analysis for CDC and looking at some of the results from one of the 7 groups that were providing them with genomes, but we also did this with the entirety of everything that we could find in the databases, so about 10,000 projects. And there's no clear reason why these 2 SNPs were missed. I mean, clearly these are differences with the Wuhan reference. This one is caught, which is great. Underlying here, where because we were specifically looking at a different story, which I'll get to in a minute, we outline those mutations that are specific to Omicron. So these are very important mutations to find, and those that are specific to Delta. And this genome was not a Delta strain, so it didn't have any of these mutations. But clearly it's missing some important stuff. When we were trying to figure out differences between us and some of the CDC performers, we found some issues, and sometimes, a minority of times, they were due to co-infection or severe contamination. There's no way to really tell the difference, but in our minds, this is a clear co-infection of almost 50-50 Delta and Omicron. And so in this case, you can map out all the mutations for Delta and Omicron. Red or orange is Delta, blue is Omicron in this figure. And then Here's all the SNPs that are, all the variants that are identified by one of the performers and all the variants that we identify. And we notice that we also are not doing the best job in identifying everything, because our assumption coming in is that we should have a single genotype in a human. Obviously, that's not true. Viruses are quasi-species as they evolve to counteract your host defenses. There's always a lot of mutations and they mutate quite rapidly. And so we tried to now account for that and other issues that cropped up. The first reporting of Deltacron, this Delta Omicron combo, was not true. It was mostly due to amplicon dropout. The way things were done for this pandemic, which was a recapitulation of what we did for Ebola and for a couple of other pandemics prior, is to amplify the entire genome in various overlapping segments and then try and reconstruct it from there. And so you do amplicon-based analysis, you amplify from a sample, you mix it with 10,000 other samples, you sequence it, and then your output is, you try and figure out which barcode is which, and then you associate those back to the sample. There's sometimes the linkers that you use, the unique identifiers, there's a little bit of crosstalk. So if you order from IDT or these other companies and you want, uniquely tagged primers, normally they're slightly contaminated with 0.05% or 0.1% of all the other oligos that have been ordered in that time frame. And so you get this bleed through. Normally that doesn't matter. In one of these cases, or some of these cases it did, and that is because the primers used for amplification no longer worked for Omicron. That's because Omicron had a few point mutations in the primer region. That primer did not amplify. So the only thing left that amplified was the minute contaminants from the other samples that kind of cross-bled into this because of the way you're sorting your unique identifiers. And so that led to incorporating some delta into the middle of an Omicron sequence. And this is called primer dropout or amplicon dropout issues. So we developed this map where you can map the relative abundance of all of your amplicons, and you can see there's a dropout event at one of the locations. But this was pretty consistent throughout the pandemic. There were many different versions of individual protocols because they noticed at some point there's always one or two one variety of or one lineage of SARS-CoV-2 that kind of evades the amplification in one place or another, but they only really changed the methodology when it became a dominant variant in the population. They're like, we should really capture this, and so they'd make a new set of primers, and then you'd have to order a new set of primers, and then go back to your old settings. Of course, Deltacron was, in fact, to be seen a little bit later. So only a couple of months later, there was another publication showing that, yes, we actually clearly see recombination events between Delta and Omicron. So sometimes it's true, it's not a real signal, but other times it is real. So there is no single system that's out there to be able to track all of this. So we developed this tool that we call Ramify, which tries to find all the recombination events within the raw data. So we track all the positions of all the lineages. So we require a definition language of every lineage. And then with that definition, we know which SNPs belong to which lineage. And then we can look at the frequency under which we have both of those mutations in the same read. And so we developed this other visualization platform. Again, red is Delta, blue is Omicron. So in a perfect world, if you're sequencing a Delta isolate in a person, this is a fraction of the data. So you'd have 100% of the data that shows Omicron or the Delta mutations when they're supposed to be there, and you'd never find an Omicron mutation. That's not always true. There is sometimes random mutations or spontaneous mutations, and sometimes there's selection for some of these mutations. And you can see this in the Omicron sample where you'd expect everything to be 100% blue, but you see that some of these have what appears to be wild type or Delta-like. Okay, this is a mixed sample, and it's a confusing mixed sample. In a mixed sample, you'd expect to have 50-50, or in this case, 40% Delta, 60% Omicron throughout the length of the entire genome. In this case, we have, in addition, a couple of amplicon dropouts. And so there are known issues with the primers that were used. And in this particular case, this example is for amplicon 76. We have this point mutation in Omicron. That means that the Omicron amplicon does not get amplified, or at least doesn't get amplified as much as the Delta type. And so if you have a co-infection, you're going to have a lot more Delta signal from this, and then your Omicron signal essentially goes away to 0. Then it looks like this section here is all Delta. This top chart represents the depth of coverage. And so this gives you some idea that this one amplicon is just at a lower coverage than the rest of the amplicons. And you kind of need to amalgamate all of this information into one to be able to make this assessment. That does not play nicely with all the downstream tools that essentially still assume you are infected with one lineage. Pango does cover these recombination types, the X in their Pango lineage language, these X-type lineages, but there are many others that have yet to be named just because they haven't been frequent enough to name. So in the workflow that we have this type of output where we can define the number of reads that overlap 2 points in the genome that allow us to say if there's recombination or not, and then we calculate how many look like the parent lineages or how many look like the daughters of a recombination event. In some cases, there are none. In most of the cases, there are none. But in some people, you definitely see not only the co-infection, but you see active recombination throughout the length of the genome. Sometimes you see multiple recombination events, even within a single read. That means that in the population of genomes, there have been at least 1, 2, or more recombination events. And this is just showing some of the underlying data that recapitulate what the recombinants look like. The fold coverage of these is actually much lower. The picture doesn't show it, but if you look at the numbers, or if you can scan the numbers, they're much smaller fold coverage. But there are a lot fewer of these types of reads. Here, I just wanted to point out this one mutation position and this other one that's more Delta, and how you've got this combination of both Delta and Omicron, or it looks like the Wuhan wild type, or below it looks like one of the parents, either Delta, if you ignore the middle one. The middle one shows double recombination. So we don't need to go there. So as much as 5 or 10% of the population within a person has undergone recombination, which is more than was originally anticipated. Okay, so I'll switch a few other topics, also infectious disease related. I know I will eventually run out of time, and I'd like to get to the rest of the talk. So we had a couple of large data exploration events. You know, ChatGPT, everyone uses it. You probably use it to do some of your homework. You shouldn't. However, we want to apply this to protein sequences. This has been done by several groups now. And so we want to apply these large language models to be able to understand the language of proteins. And the language of, it's not just of any protein. You have that with these ESM models from Meta that have come out with a really nice language model to understand all proteins that at least have been deposited into some of the protein databases. But we trained ours on SARS-CoV-2. We partnered with Jesse Bloom's lab at the Fred Hutch, who's done a great job doing this deep mutational scanning. They do random mutagenesis of the receptor binding domain, looking at how it binds to the ACE2 receptor and its level of expression. So they have a couple of phenotypes, and they have all of this mutant data that may or may not exist out in the population, or may not yet exist in the population, and they have all the phenotypic data. And so we use these large language models to see if just by sequence alone, we could recapitulate what we get phenotypically. And of course we can, so we can predict even unknown sequences or never before seen sequences and anticipate how well it binds. And the rest of the data I'll scan over because I'm worried about time. And then we've used additional models on top of this to see what else we can get from these large language models. And of course, it's looking at the sequence data. So you'd anticipate you'd be able to segregate the data based on lineage. But we see a lot of subclusters in this left chart where we see these subclusters of Omicron. We do know there are subgroups of Omicron, and they've been named. there's actually 1,500 different types of delta lineages. So there are many, many different lineages, but here it just looks like a big mess. And the next goal here is to relate this, to see whether or not this relates more structurally or more phylogenetically. We'd be able to see whether or not there's specific point mutations that dictate some of these clusters, or maybe it has more to do with function. We've also applied this to not just SARS-CoV-2, which is this one very particular branch of the beta coronaviruses, but we looked at all the other receptor binding domains throughout the entire beta coronavirus family, and we can partition these in this type of T-SNE plot and look at how they partition by host. And so they're color-coded by host. The patterns, the symbols represent the type of virus. Most coronaviruses have been discovered from bats, and they're all called bat coronavirus. That's not a very accurate name. There's probably many subfamilies in there, but right now they're all colored blue. But you can see here, if you're tracking an outbreak or something and it looks like a bat coronavirus, and then you see it migrate over to something that's more akin to a different suite of hosts, you might be able to look at host jumping or spillover events from zoonotic diseases. And so that's the hope, which we're investigating now. And then, like the rest of the nation, we were very actively involved in doing wastewater sampling. And so we have Los Alamos being a small town and Los Alamos National Lab being a pretty large lab, we have our own wastewater treatment facility at the lab. And so we can surveil the lab population. And so we started sampling in mid 2021, and we were looking at the frequency of detection of SARS-CoV-2 by qPCR, but we are also doing some genomics from that. And there have been many other groups that have published that you can actually find even find the lineages before they come up in clinical settings. And so the goal for us as a national lab, thinking down the road of national security, is can we transition this very tailored, very specific amplicon-based targeted pathogen surveillance to a larger biosurveillance system. That's what's actually being called for by higher level national security people. They want a surveillance system to monitor any threat that's down the road. Yeah, I have no time to go through everything. So we have a large project just funded, looking for postdocs, to do everything from improve the method of sample collection, enrichment of potential signal, all the way through the bioinformatic processes to be able to analyze the wastewater data that's coming out the back end. We've already analyzed a lot of the SRA data throughout the world. We have a workflow. I'm going to skip through this. Apologies. I want to get through how can you identify the next threat when you don't know what you're looking for. Many of you have probably used Kraken or other centrifuge or other types of tools out there. There are many tools to do identification at the read-based level. And that is because if you're trying to assemble a single wastewater, you might assemble 10% of the data, and you don't want to make an assessment of what's in your sample if you're looking at 10% of the data. We've developed a different method that is highly specific and highly sensitive as well. Most of the other tools, even if things aren't there, it'll find it. So if you want to find anthrax everywhere, you can use Kraken, and you'll find it when you're sequencing E. coli, because there's obviously a fraction of the data that kind of looks similar with one point mutation or one error in the data, and it might wind up mapping better to. So Paul and Tracy, a former postdoc in the lab, developed this tool where we look at all the levels of taxonomy. And for each level of taxonomy, we essentially remove anything that's confusing. So anything that is found in other phyla, we remove from the genome. That is a phylum-specific database. So that database, we have one for each phyla, and the only k-mers left in that database are those that are not found in all the other phyla. We do it all the way down to the strain level. So we can get strain level specificity. Of course, there's very little genome left at that point, so it becomes less sensitive. But at the species level, you have about 80% on average of your genome. So it's not too bad. Things like E. coli versus Shigella, that breaks the rules, but that's kind of this human-based idea of what we should call E. coli versus what we should call Shigella. So that's a problem more in how we name things. And so methodologically, we can align or do any type of other analysis with this database, a K-mer approach or a read alignment approach or BLAST. You can align your reads, you can identify the number of hits that you get, but you also know exactly the position, the types of K-mers that you find and where in the genome those reside. And so you have a lot more data. And the biggest problem with most of these tools is you don't know what you're dealing with on the back end. They've all been developed. You're going to trust them and assume that they're right. They've all been developed on a mere fraction of the data. That includes our tool. And so we developed this tool, Gotcha1, at the beginning, and it's only on bacteria and viruses and archaea. That is a minor component of RefSeq, a minor component of WGS, but no one looks at the rest of the data. And so we've developed this new version with a complete revamp of the code to be able to calculate these databases across the breadth of RefSeq. All of the blue space is SARS-CoV-2 mostly, because we have millions and millions of genomes in NCBI. The rest represents like the diversity, and so we've included most of the diversity of this. This is just a pretty picture showing you. We're also including fungi. We have other eukaryotes in mind to include in this database, but that means you'd be able to reconcile K-mers or sequences, signatures that look identical throughout the realm of both eukaryotes and bacteria. Whereas if you only have a database of bacteria and you subtract human, you might still get some fungal signatures that map to bacteria, but in this way, we'll be able to alleviate this, and that's because we have a big project focused on bacterial fungal interactions. We do a lot of sampling, field sampling and characterization, and do biolog and different types of phenotypic assays, as well as microscopy, but I'll only show you the bioinformatics. So we've used our tools to do high quality assessment of what is found in fungi. We've done 16S just to see, well, what bacteria might reside in and around fungi, what are the interacting partners. It's kind of a gap of knowledge. Bacteriologists study bacteria, mycologists study fungi, very few study both. And the fungal world is amazingly impressive as a bacteriologist, I would have to say. And so we've used this tool, we've incorporated the fungi, we've looked at all the data that comes out of it. We've reanalyzed all the fungal genome projects. We're assuming that unbeknownst to the person who's sequencing fungi, they only care about fungi, they will assemble it and publish their fungal genome, and they'll kind of forget about all these small contigs that don't mean anything to them, that don't circularize with their fungi. And then we thought that maybe this would be a good way to try and find representation of bacterial partners. And we found thousands of these projects with bacterial signatures. Sometimes you have the entire genome of bacterium hidden in this, that was essentially just thrown away as a contaminant. And the fungal genome was published, but the bacterial portion was just ignored. And so we've gone from prior studies of this minute fraction of fungal diversity and bacterial diversity that were known to coexist or be correlated in field studies or in deeper analyses looking at actual interactions. And we've discovered this much wider breadth of interacting partners. The ones that where we found a lot of data for, we could actually design specific fish probes. We grew up the fungi and then we looked at bacterial presence, and we have a number of these diagrams or these pictures through confocal imaging and other types of imaging, and see the presence of bacteria within these fungal structures, within fungal. So we've augmented that view. We've looked a bit more deeply in a few of these. We've looked at basal lineages of fungi known to have these partnerships, and we've looked at comparative genomics. Some of these lineages are crazy. These two different species are completely different. If you're familiar with some of the work, maybe in the marine biosciences group, where they look at Prochlorococcus, high light and low light adapted, they're entirely different because the ones at the top get a lot of UV and have to modify, or their genomes get modified. These guys are entirely shifted. Their genomes are completely rearranged. They look nothing alike, even if they're in the same genus and even if they're in the same species. But through the investigations, we can find that these have not just happened once, they're continuously being incorporated within hyphae. And that's because we've looked at this from an evolutionary perspective, and we can see that they don't appear to be always co-evolving with the host. They do, it's just that there's also evidence of cross transfer, so acquisition later on in the evolution of these fungi. And maybe most interestingly, we've managed to find chloroplasts, which we thought at first was just a contaminant. There are plenty of 16S studies that find chloroplast signatures. They're even reported in some papers. We find chloroplasts, but we ignore them because they're a contamination. There's pollen floating around in the lab and we just sequence this. So we just thought, well, this is obviously contaminant. Some of the signal is really large. And so we just decided to do a tree with these, but they wind up being throughout the land plants, even throughout algae. So it couldn't be just a contaminant in a reagent. We see plenty of that when we do single cell genomics. And so we tried to map these, then we tried to prove ourselves wrong. We did enrichment protocols. We looked at some of the strains where we found the signal and we grew them up. And then we partnered with a company to develop all these probes that are specific to all chloroplasts. And we use those to do chloroplast enrichment. We tested it out with plants. That works fine. Our regression curve looks great when we have these spike-in experiments where we spike in chloroplasts and we can retrieve more and more of it. And then we find really good chloroplast signal from some of the fungi that have been nowhere near plants for years. We've also done imaging of this, and we've done fluorescent tagging of this, and we've found, we also fed chloroplasts to fungi. So we grew fungi up, we fed them, we gave them this fresh prep of tons and tons of chloroplasts, and we saw the chloroplasts sticking to the outside. We didn't know if they'd be ingested, but it looks like they are within the hyphae. We've grown up new biomass of fungi where we detected the signal and we actually find them incorporated. We have no clue as to function. We've done a couple of functional assays, but we don't know what's going on. But some of them have persisted in culture collections that have been in the lab for about 40 years. So they reside there. They seem to be replicated. We don't know how. We've looked at the nuclear genome, since often there's nuclear components that are required for replication of chloroplasts, but we're not positive. And then the last thing I wanted to end off with, which is another complete 90-degree shift, is on our microbiome data collaborative initiative. This is a large multinational lab effort led by Lawrence Berkeley National Lab. We take care of a lot of the bioinformatic components, so putting together all the workflows. The biggest challenge that we're trying to tackle is, how do biological researchers try and compare their data to everything else that's out there? It is almost impossible. You have to try and find through random keyword search, either in PubMed or on Google, both of which will give you different answers. You might find some data that has been generated by a group, but it's hard to know if you've been entirely comprehensive. You can try and search SRA if you're brave. SRA is very, very hard to navigate through. We've done it for years and it's challenging. So we wanted to create a system that could try to begin to standardize how we approach comparative microbiome science. That would allow you to compare different environments together or similar environments, meaning I would like to look at soil microbiome that's about 3 centimeters below the surface in a pH range of 7 to 8 or 5 to 6 or whatever, at an elevation of 2,000 feet. There's zero chance you'll be able to find any data set that's related to this. The JGI and the Environmental Molecular Sciences Lab, EMSL at PNNL, these are DOE user facilities that you can apply to get data from. They don't give you money, but they will accept samples and they'll generate a bunch of data. Many groups here and elsewhere have had the fortune of being recipients of some of these, and they can get tons of genomes or transcriptomes out. At EMSL, you can get proteomes or metabolomes. You can also get imaging and other things from EMSL. You can also get some metabolomes from JGI, but the main point is even these two user facilities that are tied by the hip by DOE we're not communicating, even if the same sample was sent to both locations, because they have a joint ficus. It's called ficus, but they have a joint proposal mechanism where you can then split your samples. Even then, there was no way to relate the samples provided to one place to the samples provided to the other. The PI had to get data back from one, get data back from the other, and then marry it themselves, which sometimes was confusing. So now what we're trying to do is orchestrate a system where we have consistent metadata that is collected even before the samples are obtained or before the samples are provided to the user facilities. We would like to engage the broader community to make sure that even if you're not providing data through these two resources, that you're still collecting the metadata that would allow you to compare with all the data that we're collecting and to submit it to the portal. We have three main products. One is this submission portal, and that is really a metadata portal. It's to provide all the standardized, uncontrolled vocabulary in an ontological framework for your environment and anything related to the sample. So that is a work in progress still, but we have the beginnings of it. We're capturing what we consider minimal information, which is based off of the Genomic Standards Consortium. And we're trying to implement that. We're developing an app so that if you go out in the field and you're collecting stuff, that you'll be able to plug and play or put everything as you're grabbing the sample and get automatic geolocation and other things. And so that will be upcoming. We have this edge bioinformatics based platform on our system, or based off of our system. So we're incorporating all these standardized workflows. They're all version tracked. They all are containerized. And in our system, to be able to submit to the site where you then can search for those samples, everything is processed in an identical fashion. So even if it's not the right way to process your sample, we only have one way to process these metagenome samples. So we're assuming, or we're doing this primarily for shotgun metagenome, shotgun proteome, shotgun metabolome. And as long as you have all the right metadata information as to how it was processed, how it was sequenced, et cetera, it'll automatically grab that, shove it down the right workflows, do everything from raw data, QC, assembly, read-based analysis, assembly-based annotation, assembly-based mag generation, and then provide all these data consistently. And all those data can be slurped into the NMDC portal. So the data portal allows you both has an API layer, this programmatic interface where you can automatically go and grab any outputs from anything that's in the database, but also this data visualization. So you can do a search for your keywords for any type of sample, and you'll be able to see how many other samples are in the system that have metagenomes or metatranscriptomes or metabolomes that have been generated. We have a big engagement portfolio as well. So this is a picture of those. And we are actively recruiting partners from the community. So this is a community-based enterprise. It's a user facility-based effort, but it's to really engage the community. And so we have an ambassador. You know, I've given this entire talk when I probably should have.
Oh, that was not there the whole time.
Oh, no?
It just started, it just popped up like a second.
Anyway, good, because I've only been looking at the screen. We have an ambassador program. It's geared towards knowledgeable individuals who want to really get into details. They host their own workshops at their own universities or their own institutes or at ASM or other meetings, and they are the ones that are driving the use cases for others. They're getting input from the community, and then they're delivering it back to our engagement team so that we can filter through, prioritize, and then try and deliver to the community. We have an NMDC Champions program that is for the majority of you. This is to engage students to early career researchers, and that is to give you firsthand access, knowledge, how to use the system, and to be able to beta test some of the products, to be able to tell us what is missing, what else do you want to search for, what other tools or what other outputs would be useful for you, and then provide that back to us so that we can develop the right outputs and the right interfaces for you to do your work. Everything is out in the open for most of our tools, including Gotcha and Gotcha 2, the NMDC Edge workflow system. You can use the NMDC workflows as is right now. We are moving this to Google Cloud Platform, and at that point, we will lock down everything, right? And then we're hoping to have a playground area as well, where you'll have access to all the tools that we're using, but you can go ahead and change any parameters or do anything. We likely will not cater to the newest and greatest or latest assembly tool or other type of tool until we've vetted it ourselves and then decide it's worth to put into the system. But these are things that the engagement program is meant to do. We have a number of other projects I didn't have time to talk about, some on planetary protection or life on other planets. We have a gut microbiome program as well, and we're looking at the impact of the microbiome on gut permeability and diseases related to having a leaky gut, which is kind of siphoning off bacteria into the bloodstream and wreaking havoc for the rest of your system. We've couched this under this non-alcoholic fatty liver disease that we've been looking at. We also found, we've looked at different strains of Lactobacillus acidophilus. They're common inhabitants of your gut and are in yogurt and a number of other things, and they're actively used as probiotics by companies that sell it to you. But we found one strain of acidophilus that actually mitigates this high-fat diet-induced fatty liver disease versus another strain that does not. In fact, most strains do not. This one strain does. We've done comparative genomics, and we're trying to get down to the molecular mechanisms as to how this is happening and why this is happening. And then we have synthetic biology as well, but I won't go over that. I have animation, I don't know why, but here are some of the people in our group. These are the ones that get all the credit outside of my terror of a kid. And they've done everything, and those are the people that students would be engaging with throughout most of the time. And I think Olivia's mentors were Aaron and Reed, a postdoc in the group, who's done great. And then we, of course, have, this is probably a too short a list of collaborators, and we're always looking for more. And, thank you for your attention. And I'm happy to take any questions. Didn't even go over too much.
So we'll have about like 5 minutes for questions. And if you have a question on Zoom, you can just raise your hand and I'll let you know. Or type it in the chat and I'll read it for you.
Trying to raise my hand.
Okay, you want to just go ahead and ask your question?
Yeah, I think I got it. Yeah, and I don't know if my camera's working. Its little green light is not. I have a question about mobile genetic elements and how you handle them in everything you do, particularly phylogeny as well as epidemiology, because Berkeley does that too. So I just wanted to figure out how you work that into your protocols.
Yeah, that's a great question. It probably involves a little bit of back and forth in terms of details, since we deal with horizontal gene transfer in different ways for different systems. We're actively looking at horizontal gene transfer, and there are a few.
It's the chat GPT of evolution.
Yes, yeah. So there's never been a good evolutionary framework to be able to time or catalog, the frequency events of horizontal gene transfer.
Well, yeah, that's because you overlook the people who are actually doing the sequencing, and that's what's kind of awesome, because you can pick a single colony and get moves in it when you do the sequence of a single colony.
Yeah.
So it's kind of fast.
Yes, no, absolutely. So for the HGT that you're talking about, that's within species, like recombination?
Uh-huh.
Yeah. Okay. The other type of horizontal gene transfer is like complete mobile elements that move in from one species to another or one phyla to another. And that is a different type of analytical.
Well, yeah, I think people think of it only as the plasmid mediated or transformation and transduction as the only aspect of mobile genetic elements. But there's the intracellular mobility that is the recombination, the transposons and the integrons and stuff like that. And that's one of the superpowers of mobile genetic elements that in fact facilitates the ability of the plasmids too. So, well, we can talk about this at some point. So I just wanted to find out how you guys interface with what I know about like Nikos Kekritis and the gang at Berkeley. So
we know, I've known Nikos since he joined the GGI. So we interact frequently. We worked together on a tool called Genomad, which does look
that's what I was, that's great. That's a great tool.
Yeah, so that was primarily used to do viral-based discovery or phage discovery and annotation. But at the same time, it was easy enough to incorporate plasmid mobile elements.
Hunting, yeah.
And
Great,
yeah,
thanks a lot.
there are other tools out there to do viral discovery that are maybe a bit more sensitive. Genomad is a little bit more cautious in doing its finding. And then we normally do, for ourselves, we normally use some other virus finders, and then we validate using Genomad as a backup.
Mhm.
But there's no established, general protocol for that. And I want to address your mobile genetic elements like within the cell. Currently, I think it's the same as in the early 2000s. We do nothing with that. It's almost the same as the SARS-CoV-2 story, where you can see crossover events or recombination within a single sample. And your sample is just a colony. So you can look at the same type of events using our tool Ramify to look at this, but I wouldn't say it's the simplest. It have to be very prevalent in the community and the mutant.
Well, yeah, presently we use coffee and naked eyeball kind of thing, so.
Yeah, exactly, right, yes. And in the past, I remember the first, if you're familiar with some of the pathogen work, the Sanger Institute used to sequence a whole bunch of pathogens, and we were interested in diversity back in the day, not like today where we want to sequence 1,000 of everything. They had sequenced a Yersinia pestis, and I think it was reported in the addendum or the supplementary information, but they found rearrangement events at some frequency and new mobile genetic element movements in a small fraction of the data, but they threw it away essentially. And so it's not captured in GenBank or NCBI.
That's what I want to, yeah, I don't want to take any more time, but I do want to mention just what you're getting at, and that's the ref-seq.
Yeah.
And if you pull down a ref-seq and it doesn't have what you, oh my God, I can see all these things, then I get eliminated. And that's the kind of scary policy thing. Yeah, okay.
And so we work a lot with SRA, and I think what we're hoping to do with them is to maybe migrate away from using reference genomes and more migrate toward using the raw data persistently. We want persistent data at the raw data level so that you can reanalyze it with the latest and best tools, and you should be able to analyze the raw data and be able to figure out these things using the reference genomes or using other tools.
It's how evolution happens.
Yeah, agreed. Thanks.
Thank you.
All right, well, thanks everyone. That's about all the time we have for this seminar today. So let's thank Dr.